Hello everyone, my name is Miguel and I'll be presenting to you hypoxia apoptosis, promoted from hypoxia inducible transcription factor 1 alpha. Let's first dive into what the term hypoxia means. I believe that most of you guys are biology majors or biomed majors and can already break down this word. So hypo means low, oxia means oxygen. So if you put two and two together, you get low oxygen. I have an image here of an individual who has purple fingertips that is kind of fading out down to the metacarpal. And what this individual has is sulfahemoglobin. That's when a sulfur atom actually prevents the iron found in the heme group of hemoglobin to bind to oxygen. So as you can see, you can tell by the gradient of the color which regions aren't receiving the most oxygen. Now, to quantify oxygen, you could do it in either pressure, millimeters mercury, or the percent composition. And what identifies or classifies something as hypoxic is any region that is lower than the 15 millimeters mercury or 2% composition. I have here some percentages you could reference to what is normal. I have an image on the right here. It is a tumor cell undergoing radiation therapy that is demonstrated by these orange wave-like lines. And we have these hydroxyl groups in the exterior cells that's basically denoting it that has high concentrations or higher concentrations of oxygen compared to its core as you go interior. It has low oxygen. This will result as our hypoxic area or region. When you undergo radiation therapy, you want to go in many sessions because it's really just destroying the DNA of the cells it's making contact with. This more in regards to the direct effect. It's actually destroying the DNA, and then you basically are peeling the tumor apart, if you would say. But this can be problematic for the low oxygen areas because radiation therapy is actually more proficient when destroying DNA in high oxygenated levels. So when there's low oxygen, it's not as efficient as compared to breaking it down in the exterior. This would lead to tumor reoccurrence. So that led to the question, can we use hypoxia as an advantage? I know as humans, we breathe every day, and if we don't breathe or get oxygen, it could lead to cell stress and maybe induce some type of cell apoptosis. I was thinking more of an adaptive immune response, not something of the pathway similar to the kiss of death, but something more along the lines of peripherin granzyme pathway. I have a effector T cell here that's colored orange that's releasing actually perforin and granzyme granules to the target cell. I guess in this case we'll just say it's some type of cancer cell. And what the perforin does is going to open or puncture a hole into the target cell, allowing entrance for the granules to enter. And then the perforin and the granzyme will form a complex and eventually lead to apoptosis. So H1F1 alpha, or I'll be calling it HIF1 alpha, is actually expressed when there is low oxygen levels. You could actually use it as a cancer marker for cancer prognosis. Under normal oxygen levels, where there's an adequate amount of oxygen, we'll recognize alpha ketoglutarate, which can be found on the polyhydroxylase enzyme here. And in the presence of a HIF1 alpha, it will hydrolyze that, forming a binding site for the VHL complex to bind to and subsequently bind ubiquitin. And what ubiquitin does is it tags or tags it for cell degradation via proteasome. Now, under hypoxic level, as we all know now at this point, is the lack of oxygen. We'll probably take out this section here, not probably, but we'll take out this schematic of the flow here, and will not allow hydrolyzation of the HIF1 alpha unit here, thus hindering this VHL and ubiquitin to bind to this. So that means there would not be any tagging of degradation. So without degradation, the HIF1 alpha unit will bind to the HIF1 beta unit and form a dimer and enter and translate to a nucleus and initiate gene transcription of genes that trigger processes like angiogenesis. And then as we go, I'm going to refer to this photo or picture before. Angiogenesis actually promotes the growth of blood capillaries to areas that have low oxygen. This image actually showed capillaries on here. We continue on here. While researching, I came across an article where the title is Expression of Hypoxia Inducible Factor 1 Alpha Affects Tumor Proliferation and Anti-Apoptosis in Surgically Resected Lung Cancer. I have the title here broken down, but you get the gist. It's where they examine the HIF1 alpha expression in lung cancer, the association between HIF1 alpha, survivin, and C-MyC, which is tumor malignancy indicators. There was 216 patients where all their records, their clinical records and their histological examinations were fully documented. During the immunohistochemical analysis, the staining for HIF1 alpha revealed a nuclear pattern, 26.9% high HIF1 alpha, and 73.1% were low HIF1 alpha. In regards to the patient's clinical characteristics, there were no significant associations between age, gender, or smoking. The rate of the squamous cell carcinoma was significantly higher in the H1F tumors. Okay, so we have the images here. This is from the immunohistochemistry. This first one here is the hypoxia inducible factor 1 alpha. B here is the KQ67, and this one's a survivin, as well as a positive cytoplasmic staining, and the last one is the MyC. This is what they were referring to when they were discussing the percentages. And here I have a chart from the same article. It's comparing survivin positive rate and the C-MyC positive rate under HIF1 alpha expression. And here's the high, here's the low, here's the high, here's the low. You can see under the high expression that the survivin positive rate was higher. And then the same trend over here for the C-MyC positive rate. So in conclusion from this article, this study demonstrated that the HIF1 alpha expression was associated with tumor proliferation and anti-apoptosis in lung cancer via the expression of survivin and C-MyC. In this next article, I was looking for something in regards of regulation of HIF1 alpha, and this one focused on the hypoxia induced USP22 and the location site of BMI1. USP22 is actually the ubiquitin specific protease 22 and B cell specific myelin-murin leukemia virus integration site 1. And from that article, I have a figure here. It is the reduction of the number of GCS colonies formation induced by HIF1 alpha depletion was alleviated after the UP22 transfections. The increase of the apoptotic number of GCs induced by HIF1 depletion was inversed by after the expression of UP22 OE transfection. In this article, that they concluded the function of USP22 and the site of BMI1 was actually regulated by HIF1 alpha in response to hypoxia. In this next article, implications of HIF-1 alpha and the tumorigenesis and progression of pancreatic cancer. They had several points that I wanted to make that came across it. The first one, HIF-1 alpha and pancreatic cancer, pancreatic survival and proliferation. Aberrant cells proliferation is the most basic feature in tumorigenesis. Studies indicate HIF-1 alpha promotes the proliferation of pancreatic cancer through various mechanisms in RNA and is involved in the modulation of HIF-1 alpha and the carcinogenesis. High metabolism is notably a notable property of tumor cells. Aerobic glycolysis is a pathway that can produce large amounts of adenosine triphosphate, ATP, in a short period of time, and is one of the most important metabolic modes of tumor cells. HIF-1 alpha is considered a primary conditioner of metabolism reprogramming. And most pancreatic cancer patients are diagnosed in advanced stages, or even with a distant metastasis due to absence of early recognizable symptoms, missing opportunity for radical surgical resurrection. Thus, chemotherapy and radiotherapy have become the main therapies for pancreatic cancers currently, but the efficacy of chemotherapy and radiotherapy are limited by factors inside and outside the tumor cells. Tumor angiogenesis is known to be essential for growth and metastasis of pancreatic cancer, and is regulated by, once again, HIF-1 alpha at a transcriptional level and is essential to angiogenesis. And for my conclusion, back to my original question here, that can we use hypoxia as an advantage? I concluded not necessarily. There still needs to be more clinical trials, but based off all the 3 research articles we went through, it's actually more of a regulatory kind of thing, and actually stimulates more tumor cells to grow. I look forward to your questions, and I can't wait to respond back to them. Thank you.